Okay, so hello everyone, thank you for having me. My name is Giorgio and I am in the last year of my PhD at the University of Bristol with Patricia Sanchez-Barcaldo. And today we'll talk to you about sMap, which is our new program to perform stochastic mapping analysis. Stochastic mapping is one of many methods that can be used to do ancestral state reconstructions, which basically means to extrapolate the state for some sort of morphological feature for the ancestors of living taxa based on data on these extant taxa. And the advantage of stochastic mapping over other methods is that with stochastic mapping, you not only get the states for the ancestors at the nodes in the tree, you also get a posterior distribution of states for every point of every branch in the tree. And the way this is achieved is by simulating many character histories along the tree, and then discarding those histories that are not compatible with the data. So for example, this history here, you can see that B ends up in dark red when I said it should be light red. So this history is going to be discarded. And the same goes for this one here and this one here. So in the end, we just take the histories that were compatible with the data and summarize them together. So this is a Bayesian method, which means that the results that you obtain are conditioned on the data in the way that I have just shown you. But they are also conditioned on the tree that you have used and on the evolutionary model that we used for the simulations, as well as the model parameters for that evolutionary model. The reason we decided to develop sMap is because while this algorithm is implemented in many different programs, we are not entirely satisfied with the way these other programs that deal with uncertainties in these parameters. So we went on and developed sMap to address these points. First of all, the data in sMap doesn't really have to be black or white. Instead, you can supply ambiguous data, which doesn't have to be like 50-50 either. You can just set up any relative probabilities. And this is useful, for example, if you're looking at some sort of unusual feature that you find in your sample. It might be that you have a specimen which has a really weird thing that's new, but it might also be that there has been some sort of taphonomic process, the sample has been damaged. So if you are not sure whether what you're seeing is real, you can use this approach to take that uncertainty into account. And the same applies if you're looking for the presence or absence of genes in genomes. So if you don't find a gene in the genome, it might be because the gene is actually missing from the genome, or maybe you just have a genome that is incomplete. So that's the reason why you're not seeing the gene. So using this approach, you can take these uncertainties into account. For the tree in sMap, you don't have to use a single tree for the analysis. You can use multiple trees with different topologies or branch lengths. For example, they could come from a posterior distribution that you estimated using Mr. Bayes or Red Bayes or whatever. And then what sMap is going to do is to summarize the results on some sort of a consensus tree and map all the branches from the consensus tree on the trees that have been used for the simulations to determine which branches from the consensus tree are actually present in the simulation trees. Evolutionary models are a bit more complicated. So in our case, an evolutionary model is essentially a list of rates that describe how the character states change over time. So for example, if you have a character with two states, dark green and light green, you would have two rates, one rate describing the rate of change from dark green to light green, and the other rate describing the rate of change from light green to dark green. Similarly, if you have a character with 3 possible states, then you have 6 rates that describe all the possible state changes. And each one of these rates can either be fixed to 0 to disallow a certain kind of transition, or could be forced to be equal to another rate, or instead it could be estimated independently from all the other things. So by combining these 3 possibilities for each rate in your model, you can get an extremely high number of models. Furthermore, if you have more than one character in sMap, you can analyze the relationship between these characters in 3 different ways. So the first thing you can do is to just consider them as completely independent things. So each one has its own model, its own rates, and they just evolve without interacting in any way on the tree. Alternatively, you can consider them as being dependent on each other. And this essentially means that 2 characters are going to be merged into a single super character. So for example, in this case, if we have 2 characters with 2 states each, then the super character is going to have 2 times 2 equals 4 states, and the corresponding number of rates for the model. So other programs also implement models that are similar to this, But the new feature of sMap is having conditioned characters. So in a conditioned character, what happens is that you have a first character, so in this case, the green character, that just evolves in the normal way. And this is called the conditioning character. Instead, the other character is conditioned on the first character. And rather than changing normally with the evolutionary model with arrays, this character has condition probabilities that are used to compute the new state for the second character every time the first character changes. And I will show you later on why this can be useful. Furthermore, if you have multiple characters, you may want to test whether these characters are correlated with each other or not. And you can do this in sMap by doing the d-test. The d-test involves computing the statistic, which is essentially a measure of the amount of correlation between character states. And at the same time, we also compute the posterior predictive distribution of D, which is this blue curve here. Then we plot the value of D on this posterior predictive distribution. And this lets us see whether this value of D actually comes from the same distribution or not. Because if we assume that we do not have any correlation in our data, then our D should come from the posterior predictive distribution that we have computed, assuming no correlation. However, if our value of D is different, then it might mean that it's not. So it's different, it's like more extreme. It might be it's actually from a different distribution. And we can measure this by computing the posterior predictive p-value, which is essentially the area under this orange curve here. So as you can see, the farther to the right the D statistic for our reconstruction is, the lower is going to be the posterior predictive p-value, and the more significant the correlation. So as I was saying, sMap implements many different evolutionary models. So one thing you may want to ask is, how do I choose which model to use for my data? And here there are two main approaches. One is just classical model selection, in which you use a maximum likelihood estimate to compute some sort of statistics, such as a likelihood ratio test, or ASE or BSE, and then you choose the best model based on one of these criteria. Alternatively, in sMap, you can do model averaging by computing marginal likelihoods for each model that you're interested in, and then using these marginal likelihoods to compute base factors or posterior probabilities for each model. And if you compute the posterior probabilities, then you can use them as weights to average the results obtained with different models. In this way, you can remove the address the uncertainty in the model specification by using these posterior probabilities for the different models. Furthermore, sMap has a very nice interface to specify the models, which is really intuitive, I think. And at the same time, you have like a graphical representation of your model, which lets you see which parameters are forced to be fixed to a certain value or forced to be equal to other parameters. And also gives you like the source code for this model specification, which you can then reuse in different analyses. And similarly, you get the same thing for specifying the relationships between characters. So you get this nice graphical summary, which is useful because it helps you see what the model looks like, which means that it's more unlikely that you will actually use in practice a model that is different from what you are thinking about. So as I was saying before, model parameters in sMap can either be fixed to a certain value, forced to be equal to another parameter, or estimated independently using maximum likelihood or Bayesian sampling, and we have 20 different prior distributions. Another thing that you can do is to combine these last two approaches in an empirical based approach. So first you compute a maximum likelihood estimate, and then you use that estimate to inform your prior for a Bayesian analysis. And again, sMap has a nice interface for specifying these parameters. And here, for example, if you choose a Bayesian approach, you get a preview of your prior distribution, so you can actually check that it fits with what you were thinking about. Furthermore, if you choose to use a maximum likelihood approach, you will get like a real-time progress plot like this, which shows you the maximum likelihood estimate increasing as the program runs. And after you finish the analysis, we get these likelihood landscape plots. These are essentially some slices into like a 6-dimensional space, which show you all the likelihood values that have been sampled by the program. And you can use these plots to assess whether your maximum likelihood estimate is really reliable or not. Instead, if you're doing a Bayesian analysis, you get these convergence statistics in real time, which are a bit like a real-time tracer if you want. So this plot here on the right is essentially the posterior density estimates for 2 different runs. And again, these are updated in real time, so you can check and see that they are the same distribution. And similarly here, you get these conversion statistics that can be used to assess when the analysis has converged or not. We also have this wizard window in sMap, which lets you basically set up the analysis step by step, also providing tips on how to obtain the best results from your data, and having like sensible defaults for all the parameters. But sMap is not only a graphical program, we also have command line tools, starting with the sMap program proper, which is essentially a command line program that is used to run the stochastic mapping analysis. This is useful because while for short running analysis, you can just run them directly on your computer. If you are doing a very complex long analysis, you may want to use the command line version on a cluster or a headless server, but you can still set up the analysis on your computer using a graphical interface and then run it on the server. We also have this chain monitor, which again is a bit like Tracer, but it is a command line program. And this means that you can use it on a server without having to basically download any trace files or anything. And we also have this node infinite plot sMap, which are instead used to view the results of the analysis. So if you read our paper, you'll find a couple of examples of how sMap can be used. Now we'll try in a couple of minutes to show you how we have been using it in practice to study the evolution of salt tolerance in cyanobacteria. So very briefly, salt tolerance is the ability of cyanobacteria to live in environments with a high salt concentration. So we were wondering, when did this ability appear in the cyanobacterial evolutionary tree? So we tried doing a stochastic mapping analysis using the salt tolerance data, but what we found out is that there is not enough information in the salt tolerance data alone to get a reliable answer to this question. Therefore, we decided to focus on the mechanisms behind the salt tolerance in cyanobacteria, and in particular on compatible solids. Compatible solids are small molecules that the cyanobacteria produce to balance the salt concentration outside of the cell with these small molecules inside of the cell. The problem is that while we have data for the salt tolerance of cyanobacteria strains, because we know where the strains have been sampled from, we do not have this data for the production of compatible solids, just because not all of the cyanobacteria have been tested for these molecules. However, we know what genes are involved in the synthesis of these compatible solids. And since we have the genomes for a lot of cyanobacteria, we can just look in the genomes for the presence or absence of each of these genes. So the idea here was to use sMap to consider the genes that are used for each compatible solid as being dependent on each other, and then the salt tolerance character as being conditioned on the 5 compatible solids. So first of all, the first question is, are the compatible solids actually correlated with the salt tolerance? Because we assume that because they are involved in the mechanism, but we don't know if this is really the case. So we tested this in sMap using the d-test. And as you can see, the glucosylglycerol and glucosylglycerate here are the ones that are most significantly correlated. Glycine, betaine, and sucrose are a bit less significantly correlated, and trehalose appears not to be correlated at all. This is interesting because there are like biological and physiological explanations for this pattern, but I don't think we have time to go over them right now. So second question was, when did the compatible solids actually appear in the cyanobacteria? And we can answer this by plotting for each simulation that we did in a stochastic mapping, the first cyanobacterium which possesses one particular compatible solid. And the core result here is the first one here for GB. As you can see, there appears to be 2 competing hypotheses. So the most likely thing is that this GB appears sometime during the Proterozoic. However, it is also possible, although it's a bit less likely, that the first cyanobacterium with GB actually appeared much earlier on in the Archean. And this is a cool thing that you will not be able to to see if you were not doing stochastic mapping and you were just looking at like node state probabilities of the tree. Third question and main question is, was the last common ancestor or cyanobacteria salt tolerant or not? As I was saying before, the salt tolerance data alone is not enough to answer this question. This plot here shows you the probability that the last common ancestor of cyanobacteria lived at a certain time and was had a high salt tolerance or a low salt tolerance. As you can see, we have like a 67 to 43% split, which is not really conclusive. Indeed, if you have the data for the two compatible solids which are most correlated with the salt tolerance, you can see that the results actually switch, and it seems that we have a preference for high salt tolerance, even though it's still not conclusive. Finally, if we add the data for the four compatible solids which are most correlated with the salt tolerance, you can see that actually the results start to be a lot more conclusive and a lot more strong in favor of the hypothesis that the last common ancestor of cyanobacteria had the high salt tolerance. So yeah, that I'd like to thank you for listening to me. I left a few contact details in this page, and I would also like to thank my supervisor and the University of Bristol and the Royal Society for their funding.